I'm speaking today with Dr. Rinath Jeselsohn, Chair of LBCA's Scientific Advisory Board and Assistant Professor of Medicine at Harvard Medical School, Associate Physician of Medicine at Brigham and Women's Hospital, and a medical oncologist at Dana-Farber Cancer Institute. Rinath also has her own lab at Dana-Farber, where the research she'll be speaking about today occurred. And I'm Laurie Hutcheson. I'm Executive Director of the Lobular Breast Cancer Alliance. So welcome, Rinath, and thank you so much for speaking with me today about your recent publication, A Distinct Chromatin Drives Therapeutic Resistance in Invasive Lobular Breast Cancer. We are so excited to have you take this apart for us in lay terms. As you know, we have reached out to ILC patient advocates through many forums, including from our LBCA Patient Advocate Advisory Board, our LBCA Facebook group, and through private ILC Facebook groups to collect questions for you about this study. I should also add that we received many comments thanking you so much for your great research, and I want to thank you as well. Yeah. So I'm going to get started and jump in with a kind of big question. For those of us with little basic science training, or even if we've had some, too little to know much really, it would be great if you could explain in lay terms what you did in this study.
This study really was instigated by or started from clinical findings of lobular breast cancer. So despite the fact that we know that lobular breast cancers have strong expression, most of them have strong expression of the estrogen receptor, and most of them are of the luminal A type. There is evidence that they are more resistant to tamoxifen compared to aromatase inhibitors when you compare them to invasive ductal cancers. So in other words, they're relatively more resistant to tamoxifen, which raises the question, why are they more relatively resistant to tamoxifen when they have such strong estrogen receptor expression? The other finding from studies from the past few years ago showed that lobular breast cancers have a higher prevalence of FOXA1 mutations, and FOXA1 is a key protein or pioneer transcription factor, and I will go through more of what FOXA1 is later on, but there are more mutations of FOXA1, which is a key component of the estrogen receptor transcriptional axis. So putting this together, both the evidence of relative resistance to a specific endocrine treatment and also increased alterations in a key part of the estrogen receptor axis led us to question what is the estrogen receptor axis in lobular breast cancer, or are there any unique features of the estrogen receptor axis in lobular cancers versus invasive ductal cancers? By interrogating this, can we learn more about the biology of lobular breast cancers? So that was the goal of this study. And in this study, we used mostly preclinical models. So we used 2 cell lines that are known lobular breast cancer cell lines, and they have the classic loss of E-cadherin. And we used 2 invasive ductal cancer cell lines that are used frequently in laboratory-based research. We also used cell lines that were isolated from a patient who has metastatic lobular breast cancer to confirm and validate our findings in those cell lines as well. And then some of the findings we've also validated in patient samples that were already published, and this is from the TCGA cohort. And through this work that we did mostly in cell line studies, so preclinical studies, we found that indeed the lobular breast cancer does have a unique estrogen receptor axis, meaning that the estrogen receptor in lobular breast cancer binds to the DNA in different sites in lobular breast cancers when compared to invasive ductal cancers. And this leads to transcription of genes that are different in lobular breast cancers to invasive ductal cancers. And we found that at least in part, these differences are due, or in a large part actually, are driven by differences in FOXA1. And I will go through that as well in more detail, how that happens. So in summary, really to just what the main points were is that we found that really lobular breast cancers have a unique estrogen receptor transcriptional program, and this is driven by unique FOXA1, and this leads to the transcription of different genes that could have actually an impact on outcomes and a detrimental impact on outcomes. And also through this work, we identified a mechanism by which lobular breast cancers are more resistant to tamoxifen.
Can you explain what a chromatin state is in lay terms?
Sure. So actually, I'm going to share a cartoon with you to kind of help give just a very high level overview of what is the chromatin state. So DNA is really a three-dimensional complex of both the string of DNA that is wrapped around proteins that are called histones. And this complex of DNA and histones is really what we call a chromatin. And as you can see here in this cartoon is that there are some regions in the chromatin where the histones are packed together, or this is what we call closed chromatin. And these areas are less likely to be transcriptionally active. Whereas on the other hand, you have areas in the DNA where the chromatin is less compacted and more open, or we call open chromatin, and these are the areas where transcription can happen.
Can you explain how tamoxifen interacts with the ER receptor and how FOXA1 interacts with it as well?
Sure. So in general, estrogen receptor, it is activated when it binds to estrogen, and that causes an estrogen receptor to translocate to the nucleus, where it binds to DNA. And when it binds to DNA, other proteins bind to the estrogen receptor. These are what we call the co-regulators. These proteins can either activate estrogen receptor or they can inhibit estrogen receptor. And the complex of the estrogen receptor together with its co-activators basically will promote the transcription of many, many genes. And this is because or the estrogen receptor, it doesn't bind to one site in the DNA, it binds to thousands of sites in the DNA. And by that, it can regulate the transcription of thousands of genes. So when tamoxifen basically binds to the estrogen receptor at the same site that estrogen binds to the estrogen receptor, and it basically blocks the binding of estrogen to the estrogen receptor. So it competes with the estrogen to bind to estrogen receptor. And basically what it does is it changes the balance of the binding of co-activators to the estrogen receptor, and basically leads to a decrease in co-activators and an increase in co-repressors, and basically leading to the repression of the activity of the estrogen receptor. And this binding of tamoxifen and the balance between activators and repressors is very specific to the cell. So different cells also can behave a little bit differently when tamoxifen is bound to the estrogen receptor in them. Now, the FOXA1, what it does is, as a pioneer transcription factor, is that it doesn't necessarily bind to the estrogen receptor itself. It can, but it can also, basically, we know it binds to the DNA in the same region that the estrogen receptor binds. And what happens is it binds to the DNA, and it has these wings or forks that basically open DNA. And what it does is it opens the DNA, and that enables the binding of the estrogen receptor. So that's why it is a key transcription factor, or what we call pioneer factor. And it has a role in dictating the chromatin state, because it can lead to sites that are open or closed, because it can actually open DNA.
How is the presence of the FOXA1 interfering with what tamoxifen is doing?
So what we saw was that in lobular breast cancers, we had a lot more of FOXA1 binding, and that led to a lot more sites in the DNA that was open. And we saw that in those sites that had unique FOXA1 binding, we had continued ER-directed or ER-mediated transcription, even with tamoxifen. So what it looks like, what we saw was basically that the increased binding of tamoxifen blocked the inhibition mediated by tamoxifen.
Another question is about a statement in the publication. It says ILC may have a unique ER axis compared to IDC. Can you explain what that axis means and how it relates to what you just talked about?
Yeah, so basically when we say the ER axis, we mean all the sites that the the estrogen receptor is binding to DNA, and the genes that it is its target genes, so the genes that it's promoting its transcription. So that's the estrogen receptor axis. And basically, because FOXA1 binding is different, or we have sites that are gained or are unique to lobular breast cancers where we have FOXA1, that opens DNA in specific regions unique to lobular breast cancer, allowing estrogen receptor binding. And in turn, this allows the transcription of genes uniquely in lobular breast cancer when compared to invasive ductal cancer.
I guess this is sort of you're really explaining the process of the change in the receptors that then result in a resistance to tamoxifen. And when you talk about silencing FOXA1 in the study, resulting in the loss of 6,215 accessible sites, is that related to this process in the same way, or does that mean something different?
Yeah, so one thing that we did as a validation in our study was really as another level of validation was to silence FOXA1 and show that many of those sites that we found have FOXA1 binding and are gained in lobular breast cancers. If we silence FOXA1, we'll lose those accessible sites. And we didn't see loss of all the sites, but we saw loss of many of those sites. And that also raises, to us, that raised the questions that if you have a site that already is open, and even though it's dependent, the opening was dependent on FOXA1 binding, if you silence FOXA1, what's the timing, or is it already open? Will it close? So we didn't see full loss of those open sites, but we saw loss of many of those sites.
And I'm assuming that sort of will relate to what do we try to silence FOXA1 and how likely will it have an effect to reverse that resistance, right?
Yeah, though we have to keep in mind that in the lab, you know, in general, many of these experiments are short term, and obviously when we're thinking more in the therapeutic, in therapeutics, it's more long term. So those are type of experiments that we would need to do more long term or a time course.
Similarly, would you expect that this resistance mechanism that you found in this study also be a mechanism of resistance to other endocrine treatments in lobular breast cancers?
Yeah, so I think that this is probably more unique to tamoxifen, and that it actually, in some ways, it actually supports better, trying to better inhibit the estrogen receptor in lobular breast cancer, because it has it's a unique drivers that are dependent on estrogen receptor, meaning that potentially some of the novel or new endocrine treatments which cause estrogen receptor degrader might be better in lobular breast cancers.
So would you recommend or yet that patients with ILC test for FOXA1 levels or FOXA1 activity? And if so, what kind of test would that be?
Yeah, so at this point, this is very still in research and investigational. We don't know, is there any treatment that we can we change our treatment based on FOXA1 alterations, and that will in a manner that will improve patient outcomes. So until we have more data on in patients, what is the meaning of these alterations, and what can we do to utilize this information for clinical purposes, I would not recommend to test this on a routinely based as a standard of care.
So you're talking about a need for next having some clinical trials.
Yeah.
So you mentioned in the article that FOXA1 is an undruggable target. Can you explain what that means? And does it mean it's a potential biomarker, but that more research is needed?
Sure. So one thing, it is a potential biomarker, but historically it is considered an undruggable target. So it isn't as opposed to the estrogen receptor that is bound to estrogen. So there is a domain in the estrogen receptor where we know a different compound can, or protein, or can bind to the estrogen receptor. The FOXA1 does not have a ligand that binds to it, so there's no necessarily known docking site for a drug to bind to FOXA1. That said, we and other groups are interrogating FOXA1 very closely to see, are there other ways to target FOXA1. In our study, we identified a mechanism to silence FOXA1 by targeting the enhancer that mediates the actual transcription of FOXA1, and we targeted a very unique enhancer that we identified and led to very profound silencing of FOXA1, and also profound silencing of tumor growth, supporting the silencing of FOXA1 in general. And that could be a potential mechanism. Right now, this is really preclinical work, and definitely needs a lot more work in order to bring a type of drug that silences the transcription of FOXA1 to the clinic.
Okay, great. I think I understand. I understand it's not a druggable target, but silencing the enhancer would be a druggable activity.
Right, that could be a mechanism to target FOXA1, but there can be other potential mechanisms, such as FOXA1 might have a specific phosphorylation site. So that's a post-translational modification on FOXA1 that is critical for FOXA1 activity. And if we have a protein that can silence, or a small molecule that can silence the phosphorylation in a specific site, that could be a way to target FOXA1. So there could be multiple mechanisms to target it, but historically we haven't been able to target it. That doesn't mean that in the future, as we do more work, we won't be able to, hopefully we will be able to target it.
Yeah. Well, so that leads to, what are you going to do next? You've noted that there needs to be more ILC models and more clinical trials. And where do you think the next step, or is it simultaneous research could happen?
Yeah, so I think there's simultaneous research that could happen. Right now, we are working on analyzing clinical samples from a clinical trial that we did here at Dana-Farber. Otto Metzger, my colleague, was the principal investigator on this study. This was a study where patients received either tamoxifen or an aromatase inhibitor before surgery, and we have clinical samples from before treatment and after treatment. And now we wanted to look at the estrogen receptor axis in these samples to see if we really see what we saw in this study. So we want to validate it in a relatively large patient cohort. And also, I think now that there are novel endocrine treatments in clinical development, it will be really important to study in clinical trials which one of these, or maybe not just one, but is there one or a combination of endocrine treatments that might be uniquely or specifically better in lobular breast cancers?
So it sounds like, and correct me if I'm wrong with the term, that you could add a corollary study about FOXA1 to any one of these new trials of some of these new endocrine treatments that are being tested.
Yeah.
Yeah. And does that mean you have to go and ask, or like, is there a way that the Lobular Breast Cancer Alliance or advocates can help, sort of promote this and make it happen?
Absolutely. I mean, I think, one thing is obviously these studies have a cost to them, unfortunately. Yeah. So having resources to do these type of correlative studies would be important. And I think also, advocating participation of patients with lobular breast cancer in these studies. There are a lot of ongoing studies looking at the novel endocrine treatments. So participation of patients with lobular breast cancers, I think, will be very helpful and revealing. So those are things I think we could, that LBCA could be very helpful in promoting.
That sounds fantastic. And I'm wondering if there's anything we didn't ask you that you think it's important for us to hear about, or anything that you want to reiterate about what you just shared.
I think one thing that our study highlights is really the unique biology of lobular breast cancer, and I think we need to continue to investigate what are really the fundamental unique biology. What is the unique biology of lobular breast cancer beyond what we know today?
Yes, absolutely. Thank you. And thank you so much for this research and for taking the time to share.
Thank you for having me.